Hello, and welcome to Quantitative Skills of Graphing. So when we collect data, there are two types of data. The first type of data is qualitative data. This is really non-numerical data, and it's usually subjective. So for instance, we can go to an event and we can say, this was fun. Now, there's no numbers obviously involved with that. The second type of data is quantitative data. This is where we have numbers. It allows us to do statistics. It is usually data that we measure using an instrument, such as a time would be measured with a clock, or temperature would be measured with a thermometer. Quantitative data can either be discrete or continuous. Discrete data is ones which have a definitive number. These are usually integers. And what this allows us to do is it allows us to take these data points and put them into buckets or categories. The other type of quantitative data is continuous. So this is basically an infinite number of values. A lot of the time this happens with a lot of decimals, and what this does is it allows us to form a continuum. So if we think about human height, some people are short, so they could be say 4.5 feet tall, and some people are very tall. They could be 6.5 feet, or anywhere in between. And we'll see later that that ends up being a bell curve. So the first thing I have to ask you is which graph would represent continuous data and which graph would be discrete data? Think about it. Graph B is discrete data. You can see that we have 4 different groups. Group 1, Group 2, 3, and 4. And so when we collect those data points, we put them in those bins or categories. Whereas Group A, which measures the time of growth in days, we can have data points anywhere along that time scale. So graphs are used in science, but they're used almost in any other subject that you may study in the future. And graphs are important because what they do is they allow us to take data and they allow us to organize it in a visual manner which represents the data and allows us to compare the data points. This would be an example of a scatter plot. On the y-axis, you can see grades, so that was the grade that the student achieved, and on the x-axis, you can see the hours that the student studied. And sure enough, you can see a direct correlation by the hours that a student studies by the grade that they achieve. Hint, hint, hint. So next we're going to look at a couple of different types of graphs. And we're going to start off with the line graph, and that's probably the most common one that you are familiar with. Now we usually use a line graph when the data that we're plotting on the x and y axes is continuous. The dots on the line graph indicate measurements that were actually made in the experiment. A line graph is usually data that we collect over time. So say for example, if I ask you to measure the temperature for every day this month. So you're measuring the same thing. You wouldn't measure the grades that students got on a test using a line graph because each one of those data points is unique for that student. Alternatively, you could use a line graph to graph the progression of a student over the year. So for instance, you could graph their test scores on each test that they receive, and hopefully you would see an upward trend for that student's progression in the class. So what are some traits of a good graph? Though a good graph has a title, and I will tell you that this is something that students don't do well at. So what defines a good title? Well, a good title is one that tells exactly the information the author is trying to present with the graph. So a couple of examples of graph titles could be the relationship between study time and score on a biology exam in 2011. So in many examples, what we usually do is we take the independent variable, for this instance, time of studying, versus the dependent variable, which would be the score on the biology exam. So that's an easy way to write a good title. The axes should be consistently numbered. So you can see in my example, I go 0, 20, 40, 60, 80, 100. Don't make these weird jumps, like for example, 0, 25, 30, 35, 50. Keep consistent. The axes should also contain labels in units. And when I grade your assignments or when I grade your tests, that's an area I always take marks off for students. So they should be descriptive. Also, they should have units. So for example, time studied, well, is that minutes, hours, days? Let me know what that means, okay? So make sure you have both the unit and good descriptors on the X and Y axis. This is an area where I always take points off on students' assignments. I want the independent on the X axis. That is customary to do, and we always put the dependent variable on the Y axis. Once again, that is the customary way to do that. So what that means is whenever I as a scientist look at your graph, I know immediately what you changed and what you measured. Consistency is the best. And lastly, the line should not be extended to the origin if the data doesn't start there. So a lot of the times in your graph, the XY origin is usually the 0 point. You don't necessarily have to put your data starting there if 0 is not the starting point. So just be aware of that as well. Extrapolation is the prediction of what the chart might look like beyond the measured set of data. If I asked you, well, what happens if a student were to study 4 hours, 5 hours? What would their grade be then? So what you would do in that situation, you would take a line and you would extend it based upon the data points and predict what would happen. A lot of the times what we do is we indicate the prediction with a dashed line. And what that suggests is that we actually didn't measure those data points, but based upon our data thus far, we would predict that that's what we would see, the slope of a line. Slope is rise over run. Question asks you to calculate the slope of the line. Find y2 minus y1, subtract x2 minus x1. And my suggestion to you is pick points that are easy. Don't pick ones you have to guess. A positive slope is one that is going upwards. The negative slope is one that's going down. And then zero slope, you can see, is going to be a flat line. The next thing you'll notice there at the bottom of my slide there, those little dash marks there, that broken axis, we do that if we skip some values. So notice in this graph right here, our data points start at 23 and go up to 34. It just doesn't make sense for me to draw all the way from 0, 1, 2, 3, 4, all the way up to 23. So rather than make my graph look really awkward, what I will do is I will put those little squiggly lines there to indicate that there's a broken part in my graph. And so make sure that you do the same if that's something you choose to do. So sometimes when we have multiple data sets, we chart them on the same graph. And this allows us to compare one set of data points to the other data point set. However, please be careful. You need to make a legend. So as you can see on the graph here, I have multiple data points, but you're able to distinguish the different data points based upon the color as well as the symbol. So X's or squares or circles. This may be something you'd be required to do on an AP exam. But remember, on an AP exam, you're only allowed to use a pen. So you can't use different colors. So different shapes is your best bet to do. So in biology, once we collect the data, we now need to do stats on the data. And basically that allows us to determine if the data that we collected is valid. Does it actually mean anything? And we can actually put these stats on our graphs to help support our data. So to begin with, I first want to talk to you about the difference between population and sample. Sometimes in biology, we can't measure the entire population. So think about if we were to measure the entire population of all the students at Dawson. That's like 2,500 students. Rather, a lot of the times it's easier to collect a sample. And a sample is a representation of the population. So I randomly would select maybe 25 students, and those 25 students would represent the population as a whole. When we do random sampling, we have to be careful that we don't undergo sampling bias. So back to my example of sampling the student population at Dawson. I said I was going to collect 25 students. Well, imagine if of those 25 students, all of them were seniors. That doesn't necessarily represent the student population as a whole. I'm neglecting the freshmen, the sophomores, and juniors. So when we do our sampling, we need to be careful that we're sampling a population is representative of the whole population. A normal distribution, also known as a bell curve or normal curve, occurs when our data is continuous. So as I mentioned before, the human height is a continuous curve. Some people in the population are really short. Some people of the population are really tall. However, on average, most of us are kind of in the middle of the bell curve. So when you are planning your experiment, you should think about what type of data you are going to collect and what type of statistical analysis are going to be performed on that data set. That will help you as you are collecting that data and making sure that your experimental results are valid. There are three major types of data. The first one is parametric data. This is data that fits the normal curve like we discussed before. Non-parametric data. This is data that doesn't fit a normal curve. And the last one is the frequency or count data. So in normal or parametric data, this data can usually fit in a normal curve distribution. As I said before, this data is continuous and is usually generally in a decimal form. In non-parametric data, this doesn't necessarily fit the normal distribution. It may include outliers. The other unique thing about parametric data is it actually could be qualitative data. So the last and final type of data is frequency or count data. This is generated by counting how many of an item fit into a distinct category. This can also be used to help us collect percentages. When we look at statistics, there are two types of descriptive statistics. The first type is the comparative statistics, which compares variables, the dependent versus the independent, and then we also have associative statistics that look at the correlation between the variables. So as I increase the temperature, how does that affect the time? Here are some examples of comparative statistics. Notice that in each situation, we are asking the question, is A different from B? And then the associative statistics, in this situation, what we're looking at is how are A and B. So in this instance, we're looking at temperature versus latitude. And notice how as the temperature decreases, the latitude increases. So there is a inverse correlation between those two, or a negative correlation between those two. So we talked about line graphs. Another type of graph is a bar graph. A bar graph are useful because what they do is they are used to visually compare two samples of categories or count data. The other advantage of using a bar graph is it allows us to include some statistics. Some of these statistics include error bars and means. You'll notice there on the right hand corner I have error bars in my graph. Now, I have many times seen FRQ questions that talk about the error bars. And what you'll notice there that's unique, so if you look at apple versus oranges, notice how those two sets of error bars actually overlap slightly. And what that means from a statistical standpoint, even though it looks like that oranges have a higher price per pound than apples, on average, there is those error bars which suggest that even though the average price is slightly different, by chance they're probably the same. And we'll talk more about that in class and give you some more examples of how error bars are used. The next type of graph here is a scatter plot. Scatter plots are useful when we compare one variable versus the other. So when we're comparing the measurable variable against another variable. And what we're doing here is we're looking for trends. A lot of the time I will ask you to fill in a line of best fit and see if there's a correlation between those two variables. If it looks like there's a relationship between the two variables, I will ask you to draw a line of linear regression, which is also called a line of best fit. And really what that helps us do is it helps us define what that pattern that we see is. A lot of the times we'll ask the computer to do this because it's less biased than our eyes are. The next type of graph, you may have seen these are called box and whisker plots. The purpose of these graphs is it allows us to compare two samples of non-parametric data. So data that doesn't necessarily fit that nice normal distribution. So in these graphs, the ticks at the top and bottom of the vertical lines show the highest and lowest values in that data set. The top of the shaded box represents the upper quartile of those data points, and the bottom of the shaded box represents the lower quartile. And if we had a horizontal line, that's going to represent the median in that example. Histograms are basically frequency diagrams. These graphs allow us to display the distribution of the data. You can really see a nice spread of the data, where the center there is where the average is. However, you're going to have some data points that are on the tails of the normal curve. And that really shows us the spread of the data from highest to lowest. So in order to create a histogram, what you first need to do is create bins or categories. And these must be uniform ranges that cover the entire range of the data. So if I have data that ranges from 0 to 100, and say for example I want to create 20 different bins, my bins would go from 5, 10, 15, 20, 25. Every 5 would be a new bin. And then I would take my data and I would fit them into that particular bin and graph it